So Wenxi is starting with integrating eDNA and citizen science observations to model distribution of a temperate freshwater turtle near its northern range limit. Go ahead.
Yeah, thank you, Hannah. My name is Wenxi. Thank you for hanging around at the end of the conference. I just want to make a quick announcement that we are turning into community observations instead of citizen science, but just to clear that up. So I would like to start with acknowledgement. This is part of my PhD work. My committee members, Professor Wang, Professor Cumming, and then Gregory Bote, a great collaborator, and then all the help I got, either physically or psychologically, from Lockheed Lab. So Steve's there. So I'll start with this very broad assertion that all species have their range limits, and not species are fully cosmopolitan. They all have their reasons for their limits. So for sometimes the mechanism behind the range limit is quite obvious, the geographical barrier, but sometimes it can get very, very complicated when you're looking at the definition of a range limit. So that's not surprising that for the turtles in North America, we have 8 species of turtles or 9 species, depending on how you define penny turtle, but all the turtles in Canada reach about 45 degrees north in the range limits. And to understand the limits of a certain species, that the actual understanding of the distribution is very important, because if you don't have a clear understanding of the distribution, the limit comes from nowhere. So that is not surprising for a lot of cryptic species. If you don't have a well-documented distribution, all the things I'm going to talk about will be making no sense. But today we're going to look at one species of turtles that has this cryptic life history in Canada. That is the common master. I think nobody really talked about this turtle in this conference, but this turtle, to the non-turtle folks out there, is one of the smallest freshwater turtle, one of the cutest. It's about the size of a hand. And then, as the photo show here, taken by Nick a couple of years ago, it lacks a basking behavior. It doesn't really come up to a rock or to a log to bask as other turtles. So it's entirely aquatic, almost entirely aquatic. Then to add up to the complexity of detecting the turtles just by randomly looking at from a boat, is typically confined to one water body. It doesn't really travel from lake to lake. So on the road, you don't really see this turtle. During my entire PhD, I saw them twice of the same turtle. But looking at the northern distribution of this turtle, we already have conflicting sources. So the turtle book on the top right, indicated the distribution, the range in the gray shade. Then from NHIC, that's where I consider community science observations. Natural History Information Center, they documented, curated a lot of the observation from INET. You can already see there's a disagreement with the northern range limits or the northern distribution of this turtle. And prominently, we will see 2 gaps. So Algonquin area, there's no masked turtle, and then this is probably obvious because there are farmlands and everywhere. All the wetlands have been destroyed, so turtles probably not there. But we really want to look into why turtles are here or not there. We're looking at the range limits mechanism behind the range limits and how we can use environmental DNA, this new technology, to help. So the questions break down to 3 sub-questions. So is the northern distribution of this turtle underestimated or underdocumented? And if yes, how can we use environmental DNA to help? And with this better understanding with the environmental DNA detections, what are determining factors that shape their distribution at the northern front? So for the non-eDNA folks there, again, eDNA is defined DNA, genetic material that can be directly extracted from the environmental samples. In this case, we don't really touch the actual organism we're studying. So it comes with its advantages. It's non-invasive, just as I said. It's a protected species, so we do not want to touch them. And it's very sensitive compared to conventional methods, thus is more cost-effective. And of course, it comes with the challenges, which first is that dispersion. The DNA might not be, the DNA signal might not be representing the entire water body. If you take one sample, then degradation is, of course, a very big factor. DNA only persists very short time in the water. And standardization, now all the labs are doing eDNA sort of in their own protocols. There's no global standard that we follow. But with these advantages, challenges in mind, we developed the qPCR, quantitative PCR assays to test the sensitivity, either if we can use this technology to detect turtles. So this is the Lake of Pinnacon. Tomorrow's field trip is a great place, highly recommended. So in 2 areas of this lake, where turtles are known to occur. We took all the samples, test the using the qPCR assay to test sensitivity of our assay, and we determined the detection limit is about the magnitude of 10 copies per DNA per sample. That's pretty sensitive in my book. So then with this technology developed, we set out to do field trips. So on the left side, the map here, again, I'm showing the red triangles are the NHIC records, and black dots are where I took samples. So you can see that I took samples not only from where we know turtles are occur, and also towards the Algonquin area where this turtle has not been documented at all. Of course, we're running all different kinds of trivial things. I got my truck, my little car stuck, and then a random gentleman come with a pickup truck, said, "Oh, you're screwed. I'll get you out of there." But here's just quick showing the results of the eDNA detection. So we did have the eDNA detect towards the northwest direction from Kingston, where I started. So we have some detections over there. So now with the NHIC, and also I forget to mention, there are 5 samples along the Gatineau River, way north beyond their known range. So we consider those 5 sites to be the negative control, and we did not detect any mouse turtle signal. So with the data set of NHIC and then data set from environment DNA, we start to explore how the additional eDNA detection can alter our understanding of their distribution. So that's where we use niche modeling using MaxSense. So MaxSense is a commonly readily available algorithm that relates the environmental variables and distribution of a species and trying to find a correlation. It's a correlational model. Then the advantage of this model, it takes presence-only data. That's very good for our cryptic species here, and the outputs is a prediction of probability distribution across the landscape within the constraint of environmental variables with maximum information, hence the name max entropy. And then to do the modeling, first we need to prepare the environmental variables. So just a little bit detailed on the variables we have here. So imagine you divide the map into grid, 1 kilometer by 1 kilometer grid. We have the focal cell. First, of course, we have the 19 plus summer solar radiation as the bioclimatic data, and then snow ice coverage, where we calculate how much days that this area is covered by ice and snow. That relates to the hibernation period of the turtles. Then we have elevation, because this turtle is small, the mobility, the capacity of movement is limited compared to other turtles. So we consider this elevational variables. Then, of course, we count a lot of landscape features, including the percentage of forest, percentage of water, percentage of the shoreline, that is the littoral zone of a lake. Then with this landscape variables, we consider 5 kilometer neighborhood that kind of like describe the neighboring environments. So we have another 8 variables. So altogether, we have 40 environmental variables that we unified into one big 1 kilometer grid cells. And to counter that, because we own for the all population distribution across southern Ontario, we only sample in the red area. That is where so to counter the different geographical extent, we have 2 accents to be modeled. That's in the red boundary is our inclusion of all our eDNA sampling locations. On the larger scale is the inclusion of all turtle presence in southern Ontario or in Canada. And this is the optimization framework for our modeling. So MaxSense is really easy to use, and also it has a lot of issues, overfitting, underfitting, and a lot of false conclusion from the model. I'm not going to run down all the details, but just quickly kind of blocks. So on the top left, this is our 4 modeling scenarios. We have 2 data sets, NHIC only, NHIC plus environment DNA. Then we have 2 geographical accents. That makes 4 modeling scenarios. Here we have all the parameters that we can tune, we can play with, and just make sure the model is not overfitting, it's not underfitting. Just make sure we have the best model, best parameterization of the model. Then in the middle is the iteration of optimization. We iterate over and over again to get rid of the variables that is not so important to the model. Then finally, we rank the model using a Kaiki information criterion corrected by small sample size to find the best model of the best. So that's a lot of computation practice, but at the end, we got this huge spreadsheet, one of many huge table. I'll quickly run down this table. So on the y-axis, we have the 2 geographical regions. Then on X, we have the 2 data sets. And then in the 4 quadrants, basically on the variable column showing you the ranking of the variables, how important, and then contribution showing if, for example, if you have looking at the mean annual temperature, you show the contribution of this variable in that particular modeling scenario. And then the graph, the kind of thumbnail graph is showing the response. So on the y-axis of this particular graphs, on the y-axis is the gradient of environmental variables. On the x-axis will be the predicted probability of presence of the species under that particular variable. So we're just gonna go to the bottom right quadrant of this combination data sets in the Southern Ontario region. So if you look at this, and the top 6 contributing variables, where I listed here from the shoreline area, and then from temperature and elevation and precipitation, so on and so forth. So immediately look at these are just zoomed version of the response curves. You can see those in pinkish color, those are thresholds. So we can immediately see that under certain circumstances, that turtle would not be existing. So, and then in the green shade, that we can observe some kind of optimal range of that variable that's preferred by the turtle. And if we kind of summarize them, thresholds, we have temperatures, mean annual temperature, and the warmest temperature of the mean temperature of the warmest quarter, which is summer. So we have a threshold of temperatures. Then elevation, surprisingly, that above elevation of 400 meters, the turtle will not be existing. And then precipitation in the summer, that's very interesting because we kind of suspecting because this turtle lack air basking behavior. So the ways they warm themselves out is in very shallow water. So water temperature is really important for their body temperature. If you have excessive amount of rain in the summer, that will probably lower the temperature of the water down. So that will not be good for a turtle. And then we have the optimal range that both speaks to the aquatic landscape feature that you have about 50% in a particular cell group, you have 50% water, you have 50% land. So that speaks to the interface between water, aquatic environments, and terrestrial environments. So with that, so that's one side of the modeling that tells you the importance of each of the variables in the model. And then you can also use the model to predict the relative occurrence across the landscape. It's a little bit glitched, the photo, but basically what we did is we subtract using the prediction from the combined model, we subtract with the NHS only model. We found that warmer colors indicates that our combined model predict higher presence of this turtle. That's in both cases, in a smaller geographical region and larger region, there seems to be moving towards northwest, towards north. So the range limits are supposed to be a little bit moving to the north. And down below the graph just showing we did some fancy statistics, meaning that those two distributions from combined and NHS only, they are statistically different. And then finally, if we use a cumulative output of the model, we can sort of draw out the boundary line of this. It's not substantial, but we can definitely see that in this areas I pointed out by arrows, those are my combined model will predict that turtles will be present there, and those had not been documented before. So to summarize, first, environment DNA can be an effective tool to survey cryptic animals, reptiles. The reptiles is underrepresented in the environment DNA community, and turtles. My turtle basically can be detected by this novel technology. And then distribution of the turtle in the southern Ontario, the northern distribution is probably underestimated. This needs further validation, either using more environment DNA surveys or traditional survey methods. And then finally, northern range limits of this turtle is limited by various factors, including thermal conditions, precipitation in the summer, elevation, and then aquatic features. I think I'm right on time. So with that, I would like to thank you. And we just published it this year, so scan the code. I'll leave this slide here. And also I have backup slides. If you have questions, I can probably put them up. Thank you.
Next, we have Reta Meng to tell us to not worry and be happy about blandingii turtles. Go ahead.
Yeah, so heard a lot about sad stories, and today I might talk about a little bit of a happy story. So like Hannah mentioned, my name is Reta. I'm a PhD student in the Chow Fraser Lab from McMaster University. And as many of you already know, we have 8 different species of freshwater turtles here in Ontario. And the one thing that brings them all together, beyond the fact that they're all freshwater turtles in Ontario, is that they are all considered a species at risk, federally speaking. The Blandingii turtle is specifically the species that we're studying, and a lot of other folks are studying as well. Other than the fact that they're charismatic, they're also considered an excellent umbrella species. They are considered to be threatened in Ontario and endangered in Canada. They have a rather limited and localized range, especially in Canada, and we have a small distinct population as well. They are often considered an indicator of a productive ecosystem where a Blandingii turtle can be found. You can often see a lot of highly productive wetlands, a lot of food sources, and other interactions between different organisms. That brings us to the fact that Blandingii turtles extensively travel, and they have a large amount of habitat requirements that overlap with many other species, including species at risk reptiles and amphibians, and other fish, species at risk plants, etc., which is why they're an excellent umbrella species to study. The one thing about them is that they're notoriously difficult to conserve, similar to other freshwater turtles. And this comes from a complexity from their life history trait, where they experience delayed maturity, where it takes between 14 to 21 years for them to actually sexually mature and start contributing to their population. And they experience low annual fecundity. When the females do become gravid, they usually only lay about 1 to 2 clutches a year. And within the clutches, there's usually between 6 to 21 eggs. And out of those eggs that do get laid, the ones that do survive is pretty grim. So baby turtles, especially Indians, don't really have a good time. And they experience extended longevity, with individuals in the wild being observed to live more than 75 years. So it takes a long time for an individual to get replaced within the population. And therefore, overall, their population structure requires that there be a high annual survivorship of adults and juveniles in order for a population to remain stable. Another aspect of Blandingii turtles specifically is that they're known to have a propensity for long overland travel, where bringing us to their main threats. So of course, Blandingii turtles, due to their existence in the world and the fact that there's a lot of humans everywhere, they are harmed by habitat loss and degradation, especially in highly human dense areas such as Southern Ontario. They also are experienced or are affected by land use fragmentation, where habitat may not just be degraded or lost completely, but a wetland may still exist, but be cut through by some sort of features, such as linear features, including highways, train tracks, etc., which then leads to rogue mortality, where especially nesting females are very prone to rogue mortality as they do that large migration during that time. So overall, again, it's led them to be federally endangered and provincially threatened. And they're distributed, you've seen this map before, probably. The red area is where they are distributed across North America. So you can see a lot of their distribution is in northeastern United States, actually, and unfortunately, very human dense area as well. And then in the Canadian area, we have a localized range that's mostly limited to the Great Lakes St. Lawrence region, with a distinct population in Nova Scotia. And as you can tell, based on this map and just our general geography, there are very few areas within this red area that remain low in human disturbance. So this study comes in. Based on our literature review, only 2 other studies really look at Blandingii turtle habitat use in a relatively undisturbed region, one being in Algonquin Provincial Park and the other being in the United States and Nebraska Sandhills. So our study aims to provide a baseline on how Blandingii turtles do their habitat selection, use, and move around in their landscape in a relatively undisturbed condition that fills this knowledge gap. And then once we find out what's happening, we can use this to compare between degraded and reference sites in the surrounding region. And so we predict that if a habitat is abundant and accessible to the Blandingii turtles, then the adults will use all habitat types according to their availability, therefore showing neutral selection most of the time. Our study site is located in northern Georgian Bay, and it's relatively, compared to Southern Ontario and other parts of the United States, lower in human density. And it's rarely studied with a unique Canadian shield geology that differs to much of the other areas in a Blandingii turtle range. And specifically, this study is located in an archipelago landscape with more than 500 small to medium-sized islands. That is very unique in terms of how Blandingii turtles can adapt to their landscape. And it's also located at almost the northern extent of a Blandingii turtle range. So our methods, we find the turtles, we go out in May, look around for them. Sometimes they're hard to find. And then we tag them. So we tag them with a very high frequency radio tag that weighs less than 5% of their body mass in order to not affect their behavior. And then we track them. So we do a radiotelemetry programming, and this was done in 2021 and 2022 during their active season, so between May to September, and our relocation frequency was about 2 to 3 times a week. And we had 516 total relocations across 12 different Blandingii turtles, and that's 6 females and 6 males, so pretty even sex ratio as well. Our habitats that we mainly found within this specific study site were peatlands, marsh, forest, open water, and rock barren. And then based on our observations, we found that Blandingii turtles mainly overwintered in our study site, specifically in peatlands and marsh. And then they nested in forest and rock barren. They mated in marsh and parts of open water. And then they also foraged around in peatland, marsh, and open water. And this is to know that due to observational error, we're not around the turtles all the time. So just because we didn't see them using these habitats does not mean that they're not actually using them for all of these above mentioned purposes. One thing to note, after we tracked them for 2 years, we became friends and we learned a little bit more about them. So they actually had a propensity for using more open, deeper water channel. As I mentioned before, my study site is located in an archipelago where the islands are small and kind of isolated. And what connects them are these slightly deeper water channels, and they can be deeper than 2 meters in depth. And Blandingii turtles were observed to regularly, especially during the nesting season and mating season, to travel these corridors. And then we followed them and we found them doing silly things like mating and nesting. And it was an evidence that they're able to adapt to the landscape in order to basically access new environments. So after we mapped out their habitat home range, we found that the mean home range for males was 15.1 hectares in our population and 16.2 hectares for females. And then the mean daily distance traveled for males was 55 meters a day and 53.5 meters a day for females. And this was not statistically significantly different between the sexes nor between years. So for habitat selection, some of you have already seen this in Jonah's presentation, use the same diagram. It occurs mainly at 3 different scales in literature. So there's first order, which is selection at the entire population range. And then there's second order, which is selection at the individual's home range. And third order, which is selection at the individual's location. So for our study, we focus mostly on second order and third order, which is more relevant to Blandingii turtles. And this is what we found. So using the Maley selection ratio, which is essentially just the selection ratio of whether or not the habitat used is more or less than the habitat that is available. And the line, cool. The line that's this dotted line in the center, it's a ratio of 1. So if the data is completely above or below, it shows that there is non-neutral selection. And as you can see here, we only had 1 case of non-neutral selection for forest at the second order scale, and all other selection for at the third range or the third order home range scale was completely neutral. So kind of according to our hypothesis or prediction. And overall, we found that we had really similar results to the Algonquin Park reference site with mostly neutral selection. And they also found the exact same thing of just a slight non-neutral selection for forests at the second order scale. And then compared to other literature, it often shows that non-neutral habitat selection by Blandingii turtles in sites occurred more with human disturbances. And Blandingii turtles seem to only show positive selection for wetland habitats when human disturbances are present when compared to literature. And so we're basically concluding that Blandingii turtle living in our study site appeared to have unencumbered use for all the accessible habitats on the landscape to them. And then this population is a good reference site for us to compare with other populations. This brings us to our future implications, where it essentially we're reinforcing the importance of wetlands for Blandingii turtle survival, as well as the overall landscape connectivity as shown before, where our Blandingii turtles are actually able to use all sorts of corridors, including open water use, which is not often recorded for Blandingii turtles. And then going forward, we are now directly examining an adjacent mainland population, which is in close collaboration with an indigenous community, namely Whitefish River First Nation, located in the same northern Georgian Bay area. And this specific population is more affected by human disturbances, such as roadways and decommissioned train tracks. And there are a lot of community concerns about the turtle population as well. So this will provide a direct comparison of our populations at these 2 distinct disturbance gradients, which was our goal. And then my specific future research is going to focus on weaving different knowledge systems in order to better understand and contribute to species at risk conservation. So I'm really excited to share future research with you. And I would like to thank so many people, my supervisor, Pat, all the people that have helped me out in the field. Special shout out to Whitefish River First Nation Lands Department. They've come out and tracked turtles with us for the past 2 years and made lots of friends. So it's great. Thank you.
Nick Cairns, and I think a bit of a last minute schedule switch, which is awesome. So thank you for joining, is going to talk to us about lizardsickles. Here we go.
All right, thank you. And you guys are treated to my once a year button up shiny shirt day. So thank you for participating in this festival. I first want to just recognize where I work. I've been very lucky to work in some fantastic places across Canada, but this work in particular takes place in Treaty 4 and Treaty 7 territory. And for centuries, this area has been traveling meeting grounds, hunting areas, foraging sites for many, many groups, including the Hanana, the Nahiawak, the Lakota, Siksika, as well as probably some others, because that whole area has been completely changed in the last 200 years, and we've lost a lot of cultures that have been through there. So it's a really interesting area, but it's worth remembering as well. On that note, I have lots of other people to thank. I was very lucky to get a little grant from the Horned Lizard Conservation Society. It's a fantastic little group, and they were really, really kind to support me and help me buy loggers for this project. I've had lots of help and advice from people, and I have to say thank you to my current employer, the Royal Alberta Museum, for allowing me to attend this, even though I'm not currently in Alberta. So I was going to start this whole thing off requesting that people study overwintering more, because one thing I feel that we have a real advantage in Canada is that our winters are terrible and they're very hard to live through even for people. So when you do not produce your own heat and don't have access to cheeseburgers, then it's a little bit trickier to make your way through the winter. And yet so many really interesting species do. So there's really lots of questions that we can ask that other places can't ask. But there's been so many fantastic talks on other topics as well, but there's been a lot of really great talks on overwintering. So I got rid of that slide, even though I took about the same amount of time. But I want to give you an introduction to the area. The areas where I work, most of this stuff that I'm going to talk about today, because it's really preliminary, comes from my time in Grasslands National Park when I was just dinking around on my own with a couple of data loggers and my children. I've now expanded this into southeastern Alberta, but this whole area shares a lot of similarities. It's all semi-arid grassland, so mixed short grassland, and it's extremely hot. Basically, all of our precipitation falls in these major thunderstorms in the summer, with a little bit in the winter, but most of our snow comes and just sort of blows in and then scatters around. So you don't really know how much snow you actually get, because it doesn't match what you see in your yard. If you have a fence, you get a lot of snow. If you don't have any snow. But this lack of moisture leads to massive fluctuations in temperatures, both summer and winter. So over the course of a year, we may fluctuate in 80 degrees between our highs and lows. But during the day, for example, at the end of August or in early September, we may fluctuate 30 degrees in a day. So it's a very intense landscape. And where you find intense landscapes, at least the good ones, you find lizards. And so this is the lizard that we have. This is the greater shorthorn lizard, Phrynosoma hernandesi. And it is a special concern phrynosomid lizard, so it's related to Scoloporus and some of those other really cool, very behaviorally and ornate inclined lizards, and it takes the opposite approach. But it lives in these really interesting habitats. They can live in very small areas, just a couple meters squared, but those areas are usually extremely diverse in terms of their structure, which leads to really, really cool opportunities for temperature variation. So we can find 45 degrees of temperature variation at the same moment within the same square meter of a lizard occupying it. They're almost always associated with diggable soils. And so they can live in these areas probably in large part because of these diggable soils. in grasslands, that happens to be bare paw shale. There's other types of clays and with different origins in other parts of Alberta where the species occurs, but I'm not going to get into the geology of it necessarily this time. That'll be another one. But those friable soils are what make this species capable of getting out of the winter, as you can see, or out of any sort of little moment. This one was trying to escape me from taking its picture, so it scuttled down under the dust. It's just its tail sticking out there. But they also use this to get away from winter. So they use all manner of things, we presume. There's some suspects that they might be using small mammal burrows, but I've definitely seen them preparing burrows, as this one's doing here. But we know that overwintering sites are often range limiting, or at least we suspect it for many species. We've seen research on Massasaugas and black rat snakes and some of these larger snakes that have communal burrows. And a lot of species are associated, their range limits seem to be associated with the availability of overwintering sites. But horned lizards have some very weird overwintering sites, which suggest that maybe their range limits are actually kind of weird too. Anecdotally, we know they're associated with these diggable soils, but from other anecdotal observations, we also know that they don't dig very deep. Larry Powell and Anthony Russell found that they only go about 10 centimeters down, 7 centimeters exactly. Larry dug up 2 of them and found them at 7 centimeters. And so that's kind of an odd one. The other thing that they mentioned was that they tend to be associated in areas with lots of snow. And again, when you're 7 centimeters down, but the frost level is 1.5 meters, you have some interesting conundrums for yourself. So what on earth are they doing, and why on earth would snow be important? No one's really tested the importance of snow. It was just sort of noted in one report that Larry put out, and he's subsequently working on some stuff now. But it's probably pretty important. But how might it be important? Is the snow providing them security from freezing? Is it allowing them to dig deeper? But despite the fact they're little discs with spines on them, they don't dig very well. They're not sleek like a skink. They can't scuttle in. And despite the fact that this soil is diggable, it's still quite dense. Or are they using that soil to get deep enough down, and then the snow is just minimizing the fluctuation, similar to what we see in a lot of amphibians that are freeze tolerant, where they go down, they freeze, but what will kill them is freeze-thaw cycles. So some of the methods that we worked on, again, we had access to data loggers, which was really fun. We put data loggers, well, we spent a lot of time just wandering around in the bush, well, lack of bush, and looking for lizards that might be preparing or entering or hanging out near burrows. Now, that's a lot of work. We didn't find very many, but we'll get into that in a second. And then we had occupied sites and unoccupied sites. We also then compared that to loggers placed at more stratified sites, so places on different slope patterns, places in coulees and out of coulees, and places at north, south, east, and west sites, so replicated across so that we can make a bit of a model after the fact. We then compared that, we then did some drone flights and built some high resolution drones that we can then take that data and put it into. And then every 2 weeks, I would go out, no matter what the temperature was, and measure the snow with a ruler. Oh, I forgot to mention this. We'll talk about that later. Some preliminary results. So this is what we have. We have some awesome DEMs. I haven't actually made the models out of the DEMs yet because there's just an immense amount of temperature data to collect. But that's going to be a fun one to do at some point when I have some time. We had about 40 plus loggers, many thanks to Pam bringing, and I got a couple others from the Horned Lizard Conservation Society. And what we found was that the ones that were on the surface, we did put some on the surface, some underground. The ones on the surface were obviously highly variable. They ranged from -33 to +49. The ones subsurface ranged from -16 to +26. So those are 10 centimeters under the surface. So we had these really highly variable conditions. We then talked to the park, because I worked for the park and I was talking to myself. And we were able to incorporate a mark recapture study that we had been planning. And we were able to combine that with some of the plans that we had here. The park administration actually signed off on it, and we're very happy. So now we actually have some abundance estimates that are hopefully going to get going once we revisit those sites in 3 years. So we're revisiting 3 sites on 3-year cycle. So we'll actually have mark recapture every 3 years. This is one of our lizards that we marked. We just did a 1-week revisit site. So mark recapture, come back a week, see if we, or mark, come back in a week, see if we recapture anybody. Oh, I'm going to the. So we observed 23 lizards in this area. It's about 400 meters by 300 meters, which is a decent number of lizards. However, to actually find the lizards that were using overwintering sites, we had much more trouble. We only found 4 lizards at 3 sites, so 2 lizards at 1 site. But unfortunately, those 2 lizards at 1 site were found in the spring, so we only had loggers for 2 of them. So I've grayed out my statistics because despite the fact that they're significant, that's merely just because I have so many loggers recordings. They recorded every 3 hours. So it's just a glut of data. So don't take that too seriously. Let's look at the actual data. So the occupied sites froze. We'll get into that in a second, but they essentially froze for 3 months. And the control sites also froze for about 3 months, but they froze 2 weeks later, and they unfroze 2 weeks later. The occupied sites froze later and unfroze later. The interesting thing about the control sites was that there was actually quite a bit more fluctuation. If we use a freeze thaw term for if it goes from -1 up above to +1 and then back down to -1, we considered that a freeze thaw cycle. We had 0 of those in the control sites, and we had 4 of them in the, 0 of them in the occupied site and 4 of them in the control sites. So interesting. And again, if you look at these graphs, this is a climatograph. So we have the average of the 2 sites. The blue bars are millimeters of snow, and the red is the average temperature for the 2 sites. So from this, we know that, thank you. We don't know a ton because it's a very small sample size. I have to be very careful saying I know anything about this, but it's a really neat anecdotal observation. The lizards, by my limited approximation and the fact that I often make trends from almost nothing, seem to be using more stable, snowier sites that get about the same temperature as the other sites, but do it at a slightly different pace and stay there. The most interesting and the thing that I think is the most relevant that I can actually say is that these lizards do appear to be experiencing freezing temperatures. Our lizards went down to -10.6, and they were there for a little bit. And they were, again, freezing for 3 months. They were below 0. So it's just a really interesting observation because it puts them in the same boat as common lizards, Zootoca in Siberia, as well as hatchling painted turtles. So there's very few amniotes that can do what these appear to be doing. Oh, I keep going on that. Next steps, we have to use the data that we have actually already collected, but then we have a bunch more really fun projects that we'd like to do, looking at the effects of this. What are the implications for populations, for individuals? We need to use make that model and use some of the data that we've collected. Hopefully, we'll be able to look at population effects and litters. And unfortunately, my original method tried to not touch the lizards. They don't really seem to be bothered by touching very much, even compared to most herps. But I was trying to not touch them for a number of reasons. Anyways, it didn't work. It's really hard. So I have radio trackers on them now. This is, and then the other thing that I want to do while they're working on that is sort of filling in that sort of Goldilocks idea where we have the available thermal environment underground, and then we have the used thermal environment underground, so that we understand what they're doing over the winter. And I have an example from the summer that we've already built of a little model of when we catch every horned lizard, we take a ventral temperature, and then we take a temperature in the sun, and then we take a temperature in the shade, and then we just figure out where they fit. Yeah.
This is Aidan Maloney. We've been working together a bunch lately. It's great. And we're going to hear about wildfires and wind farms. Take it away.
Thanks, Anna. Hi, everyone. I'm Aidan, and today I'll be talking about my research, which is looking to investigate both the impacts of wildfire and wind farm construction on the squamate community ecology of Central Ontario. So we'll start with climate change. So as we all know, the mounting pressure of climate change is increasing the need for renewable energy sources across the globe. One of the fastest growing renewable energies is wind energy and the implementation of wind farms. Climate change is also leading to a global increase in wildfires, which many of us experienced throughout Canada this past summer. Whether in a city or the north, the effects of wildfires were felt everywhere. Because of this, it's crucial that we understand how these disturbances could potentially affect different groups of species. A lot of past research has examined the effects wind farms pose to flying wildlife, such as birds and bats. However, there's a lack of research into how wind farm construction and operation could affect squamates, many of which, as we know, are globally in decline. We can hypothesize that the largest direct threat posed by this disturbance would be the direct mortality during the construction, which would likely be high, as many of these species unfortunately don't have legislative protection. Indirectly, we can hypothesize threats to primarily consist of habitat loss and fragmentation, as well as road mortality. While there's an abundant amount of research on squamates and prescribed fires, mainly in the States, there's a lack on wildfires, which is especially true in Ontario. Once again, we can hypothesize direct threats to be direct mortality during the active fire, and this would be if individuals can't locate refugia when the fire is actively moving towards them. Indirectly, we can hypothesize habitat loss and fragmentation, lower resource availability and/or quality, as well as less refugia, which could potentially lead to an increased risk of predation. So this chapter of my thesis is looking to understand how snake richness, abundance, and health, as well as how common five-line skink distribution trends are affected by the construction and operation of a large wind farm and wildfire. So our survey study area consisted of 4 site treatments replicated 3 times each for a total of 12 sites. So we have our control treatment, which we can treat as a reference treatment. So this is our treatment that's not affected by any of our disturbances. We have our wind farm treatment, which are sites on the wind farm itself. We have our wildfire treatment, which are sites within the footprint of the wildfire. And then we have our double impact wind plus fire, which are both within the footprint of the wind farm as well as the wildfire. So this is a typical layout of a site. So it would be rock barrens surrounded by a large wetland complex, and these rock barrens are what we would target during these squamate surveys. So surveys consisted of a combination of cover boards and natural cover. So each natural cover survey would take 1 hour of survey time and consist of flipping natural cover objects that seem suitable for both snakes and skinks. So each snake encountered was processed by getting measured, weighed, sexed, and aged. So processing each snake would take about 10 minutes, and then the individual would be released exactly where we found them. Overall, these sampling events would be minimally invasive to the snakes and would end up being really quick. When encountering 5-line skinks, we would just take some basic information without any handling, as we weren't taking any morphometrics. It was always a fun challenge flipping a skink because you would never really know how they would react. Sometimes they would literally go under your boot or crawl on your leg, or they would just sit there and stare at you, and you'd have to try to shoo them away so you could put the rock back down. So I'll just jump right into the snake data. So these are all raw data collected over the past 2 field seasons, over 20 sampling events from May to August. So each color represents a different snake species. So gray represents eastern garters, for example, and green represents smooth green snakes. So as you can see here, the highest overall abundance is our control treatment, followed by the wind farm, wildfire, and then the double impact wind plus fire. If we want to look at it a bit closer, this is a rarefaction curve, which is comparing the number of pooled individuals as well as snake richness of all 3 replicates within each treatment. So here, our X, right there, would represent the number of individuals that we found, and Y would represent snake richness, which is the number of species. So the best site in theory would be in the top right corner, indicating high abundance and snake richness. So we can see from looking at this that species richness is highest in the wind farm and wildfire treatments, while the control and wind plus fire treatments are the lowest. What is largely different between treatments, however, is the abundance of snakes, with control having the highest with about 80 snakes encountered, and the wind plus fire having the lowest with only about 15 snakes encountered. When we split the treatments up by replicate, we see an interesting result. So our control, wind farm, and wind plus fire treatment replicates are all similar within the respective treatment, which you can see as I try to group them together here. Our wildfire sites, however, are quite different from each other, with the second replicate largely skewing the treatment as a whole. So that second replicate you can see is up there in the top right corner. So the sites affected by the fire have similar richness and abundances, except for that one site. So you can see that all of these sites are the ones that are affected by fire. So they're all pretty similar to each other, except for, again, that one site that's way up there. So one hypothesis for this could be due to burn severity and the presence of refugia. So it's possible that as the fire got to that area, the weather conditions were less favorable for the spread of the fire, which could potentially lead to like a decrease in burn severity, which could potentially leave more pockets of refugia for snakes to escape to during the incoming fire. But this would require more data to truly confirm this. So these are all Shannon diversity index values for each replicate and treatment as a whole. So this index takes community richness and evenness into account, with higher values indicating a richer and more even community composition. So as you can see here, our wind farm treatment has the highest diversity index, followed by our wildfire control, and then our wind plus fire double impact treatment. So these values don't put a lot of emphasis into our overall abundance, so they don't deviate too much from each other, simply because richness was pretty similar among our treatments. And as you can see, after I ran an ANOVA with treatment as our effect, you can see we didn't get any significance among treatments. So we used the scaled mass index to estimate body condition of the two species encountered most frequently, which in our case was the smooth green snake and eastern garter snake. These analyses excluded gravid females and neonates. As you can see here, there is an insignificant difference among treatments for both species once again, with insignificant p-values. So smooth greens are insectivores and likely still had ample resources in the fire-affected areas. Anecdotally, I noticed wood cockroaches to be plentiful undercover in these areas. I did notice that I flipped the lower diversity of insects. However, I don't see why smooth greens wouldn't be able to switch to a diet primarily composed of wood cockroaches if that was what was most readily available. Eastern garters, on the other hand, as we know, are very impressive generalists. And I feel like these snakes would be able to switch to whatever was really still there, just because they are such impressive generalists, being able to exist in so many different habitat types. So I think that's probably why their body condition didn't really deviate too much, just because I think if there was anything there, the garters would be able to find it and would be able to switch to it pretty readily. Now I'll move on to our 5-line skink data. So this figure represents the number of 5-line skinks we observed over the same 20 sampling events over the past 2 field seasons. Due to the size of the study area, skinks were found to be completely absent from our wildfire treatment and one of our control replicates. And we think this is actually due to what we think is a range constraint, but we're getting some historical observations to confirm this. However, even without these sites included, we can still see a pattern. So first and foremost, we can see that the wind farm and control means are similar, suggesting that skinks can still survive on the wind farm. However, when we look at the wind plus fire double impact, you can see it's severely lower than the wind farm treatment, which suggests that it's possible that the fire did severely affect the population. But again, without the before data, I can't truly confirm this. So I'm also trying to access some historical observations to see what the population looked like in these areas before the fire hit. So I've mentioned direct and indirect effects throughout the presentation to try to differentiate the impacts squamates would have encountered during and after each disturbance. So these are 4 images taken in areas that I was surveying to try to show the indirect effects of the fire and wind farm construction. So as you can see, these images display the large differences in the macro and microhabitat. So we'll start with our control, which displays a large heterogeneous cover and varied temperature profiles. The wind farm is similar to this, with the only difference being the fragmentation from the roads and turbine pads. The wildfire areas, however, have a lack of cover and temperature profiles, with a lot of large areas of just bare rock barren without a lot of low-lying shrub and ground cover, which, as we know, snakes really like to rely on. And then our double impact wind plus fire again has the same sort of effect, but again with the addition of the fragmentation from the roads and turbine pads. So, key takeaways so far. First and foremost, it seems that snakes and skinks can persist on this wind farm even after construction. It seems that there is good enough quality habitat. However, direct mortality during construction still needs to be considered, as it was likely high, and a lot of these snakes weren't specifically looked for just because they aren't SAR, so it's still something that needs to be considered. Secondly, skink population possibly declined due to the direct and indirect effects of wildfire. However, as I said, I'm trying to compare historical and community observations to see if this is truly the case. And the wildfire impacted snakes, but it's complicated. 5 of 6 of our fire-affected sites had the lowest overall abundance. It seems that burn severity and refugia likely dictate if snakes can survive the direct and indirect effects of the fire. So this research is one of the first to assess the impacts of the construction of a wind farm and wildfire on squamate community ecology. However, more research is needed to understand the short and long-term effects in all stages of operation of the wind farm, as well as wildfire ecological succession. More research will provide data to inform future mitigation and planning strategies that are needed for the global increase of wind farm and wildfires. And thank you.
Now we've got Meg and Winand to help us understand the effectiveness of mitigation translocations for the Columbia spotted frogs.
Okay, so I believe it is of no surprise to anybody here that we are experiencing a lot of habitat loss from human development. Hopefully we won't see this in Ontario with the Greenbelt. Unfortunately, we did see this on the West Coast with the Trans Mountain Pipeline. So in British Columbia, what we do in order to help conserve all the herptiles, regardless of their conservation status, from projects as small as culvert replacement to large projects like Trans Mountain Pipeline from this human disturbance, we use mitigation translocation. Simply put, this just means that we have qualified professionals under the appropriate permits who come in, round up everybody in this case in a wetland, and move them to the closest, best, or at least what we think to be the best habitat outside of that construction zone. Now, if you've done this work before, like I have, very quickly you start thinking, is this working? Are we conserving them? And what happens, in this case, to the frog after it is moved? Turns out we don't really know, and that's kind of where my journey starts. My name is Megan Winand. I am a new face to CHS. I am a graduate student at the University of British Columbia, coming from the unceded territories of the Coast Salish peoples. And under the supervision of Dr. Tara Martin and Dr. Leanne Isaac, and alongside my incredible partners and funders who made this happen, I have had the privilege of working in Mayuk Marsh, which is a marsh just outside of Cranbrook, so fairly close to the Alberta border, trying to understand what on earth happens to these frogs after they're moved. The frog in question is the Columbia spotted frog. This is different than the rest of the herptiles we've seen here. It is a frog, first of all. And secondly, it is not at risk. It's found throughout most of BC, and a recent review that we did looking at the number of translocation projects in BC, I know it to be one of the main species that are often translocated as a result of these projects. So because we don't know a lot about the answer to this, we're starting very high level, which is, do they even survive the move? And if they survive the move, what are they doing in terms of movement and their habitat use? And you can see from my photos, I used a combination of pit tagging and radio telemetry. This is my wetland, and I would visually survey it for this entire season, looking for frogs. And when I'd find them, all of them were given pit tags, and then they were given their measurements, or measurements were taken, and then a subset of these were given radio transmitters if they were appropriate size. And then they were randomly distributed to 1 of 3 treatments. They had my control, which means they're put right back where I found them. They were either moved a short distance away, so about 1 kilometer, or they were moved 5 kilometers away to a brand new wetland. And this is just to simulate the different types of these projects, therefore resulting in the frogs being moved varying distances away. And after I'd moved them, I tried to recapture them. Try is the key word. And then I'd also, the ones that were given their belts, I would track them every 1 to 3 days, and I'd take data collection on the habitat they were using and the surrounding habitat. I finished this work 3 weeks ago. So you and I are still learning what's to come of my data. But I can tell you that I did do mark recapture over from April until August. And then I did 2 stints of belting and tracking frogs in June during their summer habitat use, and in the fall when they're hopefully going into their overwintering period. I managed to pit tag 81 frogs, and while I'm immensely proud that I recaptured 59% of them, a lot of times I'd recapture them once and then I would not see them again. That is not the same for some frogs. This is 888. This frog is a gem. I caught this frog many times, and I don't know why it understood the assignment and the rest did not. So I have to rely a little bit heavily on my telemetry data. I have 38 frogs that I belted throughout the entire season. And what I can tell you so far is that everyone survived the initial movement. Woo! 35% of them did move back if they were moved from the short distance to their original point of capture. And none of the frogs that I moved to the long distance wetland even attempted to leave that wetland, whether it was to go to another one nearby or to the one they originally came from. My story doesn't end there. I will be heading back in the spring to collect data around their breeding season, and I hope to continue this conversation with you next year. Thank you.
Now we have Jonathan Choquette to tell us about conservation translocations. Take it away.
All right, yeah, I'm Jonathan. I'm a PhD candidate at Laurentian University. I'm also a lead biologist with Wildlife Preservation Canada. Today, I will give you a very, very brief look into one of my PhD chapters, Selection and Validation of Release Sites for Conservation Translocations of Temperate Zone Snakes. So reptiles are declining globally. Naturally, with 19% of the world's reptiles threatened, it's no surprise there's a growing interest in translocations as a tool to augment or reintroduce populations of some species. However, poor quality habitat is the most important reason for translocation failure with herps. Therefore, release site suitability ought to be evaluated prior to conducting translocations. And in temperate zone snakes, conservation translocations have failed due to high overwinter mortality. So practitioners have recommended that snakes be released close to suitable hibernacula, which brings me to a shout out to Anyagi and the life zone concept. The life zone is the underground space above the groundwater table and below the frost line. So the presence of a life zone may be a useful indicator of the presence of suitable hibernation sites, and by extension, suitable release sites. But identification of life zone, of sites with the life zone rather, it's challenged by the dynamic nature of groundwater and frost levels, as well as the fact, bless you, as well as the fact that snakes are secretive when they're hibernating. So, 2 of our objectives were to first identify potential release sites for the globally imperiled Eastern Massasauga at Ojibwe Prairie in Windsor, based on the presence of a life zone. And then to validate the suitability of those release sites by hibernating a surrogate species in artificial hibernacula. So we established 4 1-hectare study grids at Ojibwe Prairie Provincial Park. We installed multiple groundwater and frost monitoring stations, and we sampled those stations weekly during the hibernation seasons of 2015 to 2018. Release sites were then identified in each grid, where a life zone of 10 centimeters or greater was observed consistently for at least 2 years, for at least 2 winters. So for example, the monitoring station in this particular figure had a minimum life zone of 28 centimeters during sampling. So we identified a release site in each grid, so that's 4 release sites in total. We also established 2 reference sites at a nearby location that was still occupied by Massasaugas. And then we installed 3 artificial hibernacula per site. So in total, we had 12 artificial hibernacula at Ojibwe Prairie and 6 at the reference location. We hibernated 113 eastern garter snakes of various age classes over 3 winters On average, that meant we hibernated 19 snakes per site. And we used garter snakes as a surrogate species because they share many characteristics with pit vipers. And eastern garter snakes are known to hibernate in the same areas as Massasaugas, even sharing burrows. So we presume release sites that have suitable hibernation habitat for garter snakes will also be suitable for Massasaugas. So we found survival overall for adults and subadults was very high, was 100%, regardless of site, but juvenile survival was lower. Because of this difference, our survival analysis was based on results for juveniles only. So a Kaplan-Meier analysis indicated that survival of juveniles differed significantly among our 6 sites, ranged from 60% to 100%. The range of survival rates, however, that was essentially at the release sites, it overlapped with the range of survival rates at our reference sites. So juvenile survival, juveniles hibernated at one of our release sites had the lowest survival. This was also the study grid where we recorded the greatest number of dry well events in our groundwater wells. So it's possible, but still speculative, that overly dry conditions resulted in relatively high mortality there. But our release sites A, B, and C maintained a life zone for the majority of sampling and supported the highest overwinter survival of garter snakes of all age classes. Survival rates at those 3 sites were actually comparable to published survival rates for garter snakes. So these three release sites appear to be the best candidates for reintroductions of Massasaugas at Ojibwe Prairie. So the next step here would actually be, beyond this research, would be to test these sites using artificial hibernation of Massasaugas. And so, a little spoiler alert, we've actually already taken that next step. And in 2021 and 2022, we artificially hibernated 24 Massasaugas at those release sites, and we observed 100% survival. So they worked. To sum it up, our results will guide the selection of release sites for Massasauga reintroductions and will guide the development of a rigorous release site selection process for reintroduction of temperate zone snakes. Thanks for listening. Thanks to all our partners and supporters.
And to round out our CHS experience of talks, we've got Andrea to assess the impacts of climate change on gray rat snake geographic range in Southern Ontario. Go ahead.
Okay, thank you. Yeah, I think anyone can hear me. Hello, I am Andrea, and today I'm going to present the proposal for one of my chapters of my PhD. And so let's get going. And so as we know, the temperature of the world have increased during the past decades at an unprecedented rate, and it's only getting warmer and warmer every day. Well, year. And so in Canada, you guys know that this warming temperature had increased even faster than the other parts of the world. And this has caused several consequences across the entire country. And as this is only expected to continue in the future, it will be very important and urgent to identify those species that will be more vulnerable in the near future, so we can advance specific adaptation and mitigation plans for these species. Species have responded to climate change in very different ways, but one of the most common observed in nature is change in distributions. Species have shifted their ranges to higher latitudes and altitudes as well, although it's not the only trend that has been observed. Some species have been positively impacted by warming temperatures, expanding their ranges, whereas others have experienced range contractions, and this has increased their extinction risk. So it's very important. The different patterns that we observe in responses are of course influenced by the dispersability of the species, the magnitude of the change, and of course the adaptive capacity of the species. So understanding how these species are able to shift the ranges or track niche conditions will be very important to or crucial to manage the redistribution of species in the future. And so here we are interested in the gray rat snake, which is a species that is distributed mainly in the United States in the east part,
And in Canada, we can find it in 2 different regions and with 2 distinct populations. The Carolinian population in the Carolinian Forest region and the Frontenac Axis population, also known as Great Lakes and Lawrence population in the Frontenac Axis. This population also crosses the border to Upper New York State, but it's completely disjunct from other populations from the eastern and central United States. These populations are in danger, or well, are classified as threatened and in danger provincially and federally, and both are considered declining. And the reasons, well, is basically habitat loss, fragmentation, and specifically for the Carolinian population, but also increased mortality by roads and persecution. And let's not forget that the species is at the northern range of limit of its range, and we have already mentioned that climate will be in the future a very important factor that it will be influencing these populations and species in Canada. And so we are asking the question, what impact will climate change have on the distribution of great rat snakes in Canada? And we're going to use using ecological niche modeling, which are very good tools for assessing the extinction risk and, of course, predicting changes in distribution in suitable habitat. We're going to use we're using the occurrence data of the National Heritage Information Center with climatic variables to predict the future distribution under different climatic scenarios. And we are interested in this because we expect that these results combined with functional habitat could help us to inform strategies to conserve the species in Canada. And specifically, we are very interested on this region and if what areas this will be important for this species to be able to move north, if that is possible. And because this is one of the most intact forest regions in this particular place of Canada. And well, I will try to present results next year and thank everyone for listening and all the naturalists and scientists that have contributed to this data.